नमस्कार दोस्तों, वेलकम टू चैनल टॉक्सिकोलॉजी ऑन बोर्ड. Today we'll discuss another DABT topic and question, recertification question. The topic will be with respect to statistics and risk model. So first two slides will read about the concept with respect to statistic risk models. Different risk models are there, like log, probit, and logit. Income and Another type are one hit, multi-stage, and linearized multi-stage. Okay, so two slides will read about the concept. Then third and fourth slide will discuss about the questions, previously asked questions on this particular concepts. So let's directly dive into the concept. So statistics risk models, no, two types are there. One is statistical models, which include both log, probit, and logit. Okay, and mechanistic models include one hit, multi-stage, and linearized multi-stage. Okay, the statistical models, they will assume that different individuals have different tolerance for a particular effector. Okay, if there is a decrease in the heart rate or decrease in BP, like that, some effect, a particular effect will be there, no? So different individuals will have different tolerances for a particular effect. That is what the statistical model will assume, and that distribution of these tolerances can be described in terms of probability distribution, that is in that particular population. Okay, log probit and logit models are based on standard sigmoidal S-shaped curve. So this is the standard sigmoidal S-shaped curve, you know. So the statistical model, both log probit as well as logit models, no, they will assume that it is the standard sigmoidal S-shaped curve for dose response. Okay. The tails of these curves are somewhat flat at low doses, therefore predict a small risk at low doses, but as the dose increases, the effect will increase. So when it comes to mechanistic models, no, mechanistic models, they will assume that the observed response is based on a given number of biological events. How many events are there in order to showcase that particular adverse effect? For example, in the case of cancer, there will be different stages will be there: initiation, promotion, replication, like that. Promotion, four stages of cancer will be there. And not only that, is it single hit, is it multiple hit, based on that also. So mechanistic models, they will assume that observed response, whatever the effect is there, no, is based on given number of biological events. The simplest mechanistic model is one hit model, which is based on the premise that a single event, there is one chemical, it's going and binding, hitting the target, or particular adenine or guanine in particular DNA. So single chemical, single event, premise that a single event is sufficient to produce the outcome. The example, one mutation can give rise to cancer. Okay, hope you are understanding the this model is very conservative, this first one hit model, and it predicts high risk at low doses. Coming to second type, that is multi-hit model, it is which is based on premise that multiple hits, more than one hit is needed, two hits or three hits. Okay, each hit follows a Poisson distribution are necessary to produce the outcome. For example, five mutations are necessary to induce a certain type of cancer. These models are called gamma hit multi-hit model. The multi-hit model is less conservative and then the one hit model, and this multi-hit model, it will predict less risk at low doses than does with the one hit model. The most widely used approaches are multi-stage and linearized multi-stage models that are currently, they are part of this EPA's default model. See, those people who are reading ISH M7 guideline, no, in that also you will come across this linear multi-stage model, you will be coming across. Those people who are doing US EPA risk assessment will be, you know, in further deriving RFC and RFD, there also you will come across this type of multi-stage or linearized multi-stage models you will come across. So try to integrate and understand the concept. These are the current EPS default model. So BMC, BMDL values are also coming from this type of thing only. The multi-stage model is based on the assumption that number of different stages are necessary for a cell to become a cancer. So a cell to convert into a cancerous cell, it has to undergo mutation, initiation, transformation, and progression. So these are different stages are there, multi-hit stages, not a single event, multi-event. There are different transition rates between the stages. The linearized multi-stage model assumes that the dose response curve is linear, like this. Okay, at low doses, This is a conservative model, and this linearized multi-stage model, it will predict higher risk at lower doses. So, so this is how the concept is. Then we will see what are the important questions which were asked in the previously American Board of Toxicology recertification question paper 2000. So the question was, which of the following is or are example of mechanistic extrapolation models? If you remember that first page, what comes under single event and what comes under mechanistic model, statistical model and mechanistic model. In the mechanistic model, we have one hit, multi-hit, like that. So the right answer is, the options are, option 1 is linearized multi-stage, option 2 is log probit, option C is gamma multi-hit, option D is Weeble. So the right answer will be in the green text, that is linearized multi-stage as well as gamma multi-hit. These two are comes under mechanistic type of extrapolation statistical model, whereas log probit comes under statistical model. Okay. So let us understand from this question only little bit about the concept of what is this mechanistic models, little bit in detail in next one or two slides. So mechanistic extrapolation models are mathematical frameworks which are used in risk assessment to estimate the low dose impact, how that particular chemical is behaving at low doses, low dose impact based on biological understanding of the disease progression, rather than just fitting into a curve to data. Like in the case of conventional type of dose response, S shape curve will be there, no, where you're taking control with 3 doses, you will end up with a S-shaped dose response curve. Instead of that, we are going with this type of applying mathematical framework. We are not just trying to fit, but the particular information fitting a curve to a data. We are trying to understand the biological understanding of this, how the disease is progressing at various stages. The linear multi-stage or gamma multi-hit and related models assume a sequential process. You have seen in the case of cancer, no, initiation, promotion, mutation, like that we have seen, no. So related models assume a sequential process where a cell must endure multiple hits, okay, or stages before becoming malignant. So we'll see three different types we will try to learn from this video only. So first one is linear multi-stage model. Okay, it is called Armitage or Doll. This model assumes cancer is a multi-stage process resulting from a sequence of genetic mutations or stages that a cell must go through. So we will see different examples. What type of examples will come under linear multi-stage model? Example 1 is smoking and lung cancer. So the association of the smoking with the effect is cancer. How it comes under multi-stage, linear multi-stage, we'll see. This linear multi-stage model was used to show that lung cancer rates depend on the duration of the smoking. How long you're smoking, for 1 year, 2 year, 3 year. Okay, duration of smoking and the dose. How many cigarettes per day? Okay, this called chain smokers will be the, no, consistent with the multi stages of cancer is induced by smoking. So this is a case example which where smoking and lung cancer is a type of linear multi-stage, duration and number of times the cigarettes per day. Okay. Second example is EPA Carcinogen Assessment. They also use this multi-stage model. We'll try to understand the concept. The US EPA historically used linear multi-stage model as a default to calculate upper confidence limit. As I told you, US EPA UL value, as well as for vitamins and hormones, as well as US EPA, this RFC, RFD values will be there for inhalation, concentration and oral dose RFC and RFD. So to calculate this particular upper confidence limit, this EPA was using this particular linear multi-stage model uh, to calculate upper confidence limits on low dose carcinogenic risk from animal study data. So this is also comes under linear multi-stage model. Third example is benzene risk assessment. Apply to data or workers exposed to benzene to estimate the probability of the leukemia. So the association of this benzene exposure to the blood cancer, that is leukemia, allowing for the intermediate stages of damage, allowing for linear extrapolation at the low doses. So this is also a typical example of linear multi-stage model. So now we'll see what is gamma multi-hit model. Okay, this particular gamma multi-hit model assumes that a single target cell must receive a specific number. of that number is fixed, number k of hits from a toxin to become a active or tumorogenic, that particular chemical to become active or tumorogenic, okay, that particular target cell. Example one is radiation damage. The generalized this multi-hit model, no, it will act as a mechanism informed approach for radiation induced cell death. It suggests that this gamma multi-hit model suggests that damage is related to the probability of the lethal events, how many times the hits is occurring from the radiation exposure, how many times that particular individual is exposed to that particular radiation, and it is used to predict the cell survival curves, cell survival curves, and the effect of this dose fractionation. So radiation damage is comes under gamma multi-hit type of damage. Next we have example 2, that is toxicity data extrapolation. This gamma multi-hit model has been used for low-dose extrapolation in the toxicity studies, where the response is assumed to be the result of specific number of hits. How many number are there? Here you have seen K, which is a fixed number, typically yielding 3 parameters for the fitting. So that comes under gamma multi-hit. There is another model is their third type, that is cell kinetic models. So these are more complex versions, incorporate biological factors like cell division and death will also involved because second one was mechanistic type. This one is cell kinetic type of model. Okay, so this type of MBK model, if you see, it is a two-stage stochastic model for carcinogenesis. It will incorporate cell kinetic parameters like rate of cell division, rate of death, in addition to the mutation rate. So till now, other two were cell kinetics was not involved. In this cell kinetics, along with the mutation rate is involved. So what is the application of this cell kinetic model is to describe the time to tumor formation in epidemiological studies for leukemia as well as for the lung cancer. And if you look into this GMH, that is generalized multi-hit model for cell survival, it is used in the radiotherapy, as you can see in the case of previous slide also, for radiation to describe how cancer cells are killed. It assumes a limit on the amount of potentially lethal damage a cell can repair. So there is a, if you look into this breast cancer and metastatic prediction also, no, it is also following, it is this particular model is also relying on intrinsic mechanism of mechanistic progression, growth, that is alpha, and dissemination mu, used to predict the relapse risk. So if you see the comparison, no, model type, exam point of view, what you have to remember is model type, which type you have to apply. So if it is linear multi-stage, there is mechanism is sequential mutations, and there are different stages is there, and best application is long term chemical carcinogen, example is benzene. Coming to gamma multi hit, multiple hits to a single target or single cell, and here the best application is acute toxicity of radiation induced damage. Okay, and MVK model is clonal expansion, mutation plus growth, age dependent cancer induced studies. Okay, so these are the things that you have to remember from the exam point of view. What they will ask is, particularly you read ISH M7 guideline also. In that also linear multi-stage model, this TD50 type approach will be there. No, that you have to read. So these models are often chosen over simple linear extrapolation model because they reflect the understanding that biological systems can repair damage, and that carcinogenesis is not a single step event. That's why you have to understand the mechanism of how the cancer is taking place at multiple stages will be there, no, prolonged event. That's why we are at least doing 2 year mice and rat study in order to understand, because the cells will undergo pre-neoplastic lesion, then tumors will form. Certain tumors might be initiation, progression, proliferation stages will be there. Certain tumors might be benign or malignant will be there. So it is a a chronic period it takes for an adverse for a carcinogenic effect to be seen. Okay, not a single step event. So in that case, you have to apply this type of mechanistic models. You have to apply in order to interpret the data. Okay, hope from this video you were able to learn about multi-stage models, mechanistic models, and statistic risk models. Two questions we have seen. So direct questions is there. At least if we are aware of the concepts, what is the meaning, what is the term, and what type of study and what type of statistical model if you apply, if you are knowing that particular thing, then you will be able to address the question in the exam. Okay, thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka@gmail.com. See, thing is that please read ISH M7 guideline if you want to understand whatever I'm trying to say, or look into US EPA website. You see RFC, RFD. In that, this type of concepts they have very clearly given how they're calculating this upper confidence limit by applying this straight line curve, dose versus response. Thank you so much. Once again, all the best for your exam.